Hello students, I welcome you all to Current Affair Lectures by Azad ICS Academy, and today we are going to look into the current affairs of 25th March 2025, right? So before we go into the topics, let us look into the questions that we had seen yesterday. Oh, I guess there is some mistake. This is not the question. I'll just come up with the new proper questions in the next class, right? Okay. So the first topic for today here is what are DNA polymorphism and how do they differentiate people? So first topic is very, very important, and that is related to the DNA polymorphism. So let us understand what is the meaning of polymorphism and what is DNA. If you are not from science background, you may not be able to understand what is DNA. See, DNA is a kind of a genetic material that is present in all the human beings and all the animals for that matter. Okay. For animals, it's different. For different animals, it's different. Like for humans, it's different, and for animals it's different, and for each and every human being also these DNAs are different. So I hope all of you are able to see these lines over here, right? So one is cream color, and that is linked with brown color. One is blue color, another is linked with white color, and again you can see that the combination, the blue is always with the white and the purple, or you can say the brown is always with the cream. So there's a kind of a arrangement that happens in every human beings, and that arrangement is different. Okay. Now, DNA fingerprinting can establish parent-child relationship and help identify individuals from their mortal remain to disaster sites. DNA fingerprinting have also exonerated wrong acquisition convicts. So scientists have extracted DNA almost intact from 65,000-year-old human remains. So see, it is something which is used for forensic. So if you have heard of this word, I'll tell you what is this forensic. If you have seen any kind of a criminal movie or crime scene, this word forensic is something that's very important. Basically, what happens here is forensic team will come up with some kind of equipment, some kind of chemicals with which they can identify whose fingerprint is there. And there is a possibility that you can have a DNA fingerprint that can be found out, and by this it will help us to identify who is the convict or accused convict, or to find out whether it is correctly or wrongly present, right? So it is going to help us in paternity test, ancestral tracing, and disaster victim identification and crime scene analysis. Disaster victim identification, basically what happens is if you've lost your parents, or if you have lost your kids in the kind of what you mean to say, and if you're not able to identify, see, in the disaster anything can happen, and if you've lost your baby or if you've lost your parents, you're not able to identify who are your parents. In that case also, it can help us to identify whose parents are whom or whose children are whom, or such kind of paternity test can be also done with the help of DNA fingerprinting. So now let us understand what is this DNA first and what are these STR. We'll discuss about this. Now see, each of the individual cells. So I'll tell you, I'll just have to show you the cell structure, okay? Especially when the animal cell, this is an animal cell. Usually animal cell looks like this, okay? This is the cell membrane. And inside this, we have a cytoplasm, which is water. And here you have a nucleus, okay? Inside the nucleus, you have, something called as chromatin. And in the chromatin, you have this coil-like structure, which is called as chromosomes, right? And these chromosomes, okay, these chromosomes consist of nothing but DNA. DNA basically stands for deoxyribonucleic acid. Okay, so each individual cell in one skin, blood, like in one skin, blood, teeth, bone, etc., consists of 46 DNA molecules. So in every part of your body, see, you have to understand each and every part of your body are made up of what? Cells. And each and every cell consists of nucleus. Each and every nucleus consists of chromosomes, and each and every chromosome will have DNA. How many DNA molecules are we having? We are having 46. That is nothing but 23 sets. So if you make 23 double, it will be 46. Out of these 23, okay, one pair, that means there are two chromosomes which are called sex chromosomes. The reason is because they determine the sex of that particular person or the gender of that particular person. In males, it is called sperm, and in females, it's called egg. That's mentioned over here. You can see here. One set of 23 is inherited via sperm from the father, and another is from the egg of the mother. So the remaining 22 pairs will identify your somatic cells, they're called somatic cells, which are going to tell you whether what kind of hair you're going to have, what color of eyes you're going to have, all of that will be here. But whether you're going to be a boy or a girl, it is going to be determined by this one pair of chromosomes or one pair of DNA molecules, right? So sperm and egg are called as exceptional because they have only one copy of the genome each, not two. The fact underpin that the generation and use DNA fingerprinting to identify individual and their relative. So you can easily identify who is the individual, who is the relative. Now, the DNA is packed inside a chromosome. As I already told you, so in the chromosome, you have this DNA. So the DNA is packed inside the chromosome. For example, chromosome number 3 consists of 6.5% of the total DNA in the each cell. The chromosome number 3 is the father. From the father contains DNA that is largely similar, that is derived from the mother. A few parts are different and are called as polymerism. So what is this polymerism? DNA polymerism refers to the variation in the DNA sequence among the individuals in the population. So there are some kind of differences. So you can see DNA polymerism can be used to differentiate one person from the another person. So what happens here is the sequence of these DNA molecules are different in different people. Okay, depending on these differences, only what we will be able to understand is, okay, this DNA is different and this DNA is different. So this differences is called as polymorphism. Poly means many and many appearances, you can say, right? So they can tell us whether a parental chromosome number 3 came from the father or mother. And likewise, the maternal chromosome number 3, that is polymerism, makes the trace of ancestral possible. DNA profiles are typically generated using polymerism in the parts of DNA called as short tandem repeats. They're called as what? They're called short tandem repeats. Means what happens here is, whatever the sequence is, they keep on repeating. They keep on repeating and they are called as, and they are very short. So hence they're called as short tandem repeats, right? Now see, what happens here is it occurs sometimes. Why this polymerism occurs because of mutation, insertion, deletion and duplication of DNA sequences. Now it helps in forensic to identify the traces, etc. Now what are these short tandem repeats? We'll understand this. DNA has got two strands. Each strand is a sequence of 4 chemical bases. So please understand, I'll tell you, this is a little bit of scientific way to understand this. I'll go to this. So see, this is how the DNA looks like. Okay. And here you have these kind of things. They'll be connected like this. Okay. So basically there are 4 chemicals, you can say. If I separate it, if I make it like a ladder like this, if I remove this and this another side, it looks like a ladder. Okay. So this is one, this is one. So if you had seen the previous diagram, so here this was which color? It was blue color and this was white color and this was cream color and this was maroon color or something like that. So please understand the cream color will always be with maroon color and the blue color will always be with white color. And this keeps on happening. And then you have blue and then white and then you have cream and then maroon and this goes on happening. And this kind of repetition is different in different organisms, right? So that is what they're trying to tell us over here as well. So please understand, see, DNA has 2 strands. Each strand is a sequence of 4 chemical bases: adenine, cytosine, guanine, and thymine. So these are the 4 chemicals they may ask you in UPSC. Adenine, cytosine, guanine, and thymine are present in DNA. The strands are anti-parallel and complementary. Anti-parallel means that the strands run opposite direction. Complementary means that AS and CS of the strand bond with TS and the GS. So see, please understand, AS and CS. AS stands for adenine. CS stands for cytosine. Will always of one of the strands bond with TS and GS. So A will bond with T and C will bond with G, right? Always. When the cell division, when a cell divides and needs to make a copy of DNA, it pulls the strands apart and make a new complementary DNA of each one of them. As one strand of C gets the other is T gets the G. Thus, the daughter DNA makes the same sequence of the bases. In the rare cases, the cell may make a mistake and insert a wrong base, and this is called mutation. See, sometimes whatever I was showing you on the black screen, right? So it will be like a spiral thing and it gets separated during the cell division. And once that gets separated, it makes a duplicate of it. And during this process, sometimes, it's not always, very few cases, the wrong base may get inserted. And this process where a wrong thing is happening is called as what? Is called mutation. In general, mutation occurs less than 1 per billion base pair per generation. So every time this is happening, it's not like every now and then this mutation is happening. In that case, all of them would be mad or something like that, right? So that doesn't happen. This kind of mutation happens very rarely. Okay. Single or short tandem repeat is a short sequence of base pairs in the DNA and that repeats some more number of times, such as you can see GATC, 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 GATC. Can you see GATC, GATC is going on repeating. The four is going on repeating. And complementary strand will have CTAG, CTAG. Okay, this kind of thing is going on repeating, and this is called as short tandem repeat. They are after polymerized, unrelated, individual, almost certain have different number of repeats unless at least some short tandem repeats. So if they are not related, this sequence would get changed, right? So this is something that we have to understand about the basics of DNA and what is this short tandem repeats. Now, how do we make a use of it? See, forensic investigation identifies suspects using the DNA from the crime scene. So from the crime scene, what we do is we take this particular DNA. Okay. So scientists are making a use of this. They are making a copy of it and they're kept basically. Okay. There is something called polymerization chain reaction to make a copy of it. And even if they are very small amount of DNA to begin, we will still make a copy of it and keep it. First, scientists extract this DNA material from the tissue that contain DNA of interest and they heat it up to 95 degrees Celsius for 25 seconds and split the strands apart. So whatever these two strands are there, right? They have to split this strand. They have to separate them. And how are they separating them? They are heating it for 95 degrees Celsius for 25 seconds and split the strands apart. Second, they introduce a multiple molecule of sample. The first is the primer and the short. So this is basically a biotechnology concept wherein how they are doing this. Okay, next is another molecule of DNA polymerism is made to interact and it holds and binds the primer. And this entire process basically is going to happen. This is the process that they are explaining. Again, not important because UPSC will not ask you the process of, doing this thing, right? Just know what is it exactly and how it is done. It is done through biotechnology. Now, what is this DNA fingerprinting? A new copy of DNA is made to move in a capillary under the influence of electric field. The smaller ones move faster and technically it is called as capillary electrophoresis. Multiple strands are simultaneously sized in the same test, and the size of the various paternal maternal variants of the different SRT are compiled in the table. This table is unique for each individual and is called DNA fingerprinting. Only monozygotic twins have the same DNA fingerprinting. So please understand what is this DNA fingerprinting. What is monozygotic twins? Twins, I hope all of you understand. If the babies are born together, then it's twins. If it's one girl, another boy, then it is called dizygotic twins. Okay, if it's monozygotic twins means both of them are girls. Okay, then their fingerprint will remain the same. Okay, because it's the same sperm or the sperm had two heads, and that is the reason the cell division happened like that. In that case, the DNA fingerprinting will remain same. But otherwise, if you check my DNA fingerprinting, my brother's DNA fingerprinting, there will be slight difference. But still, you can identify that we are related. This fingerprinting is established using DNA from teeth, bones, blood, drop is also enough, spit, semen, skin, cells, etc. So, for example, if somebody is trying to do a crime, if they have taken a knife and they've stabbed on the knife, if you are able to find out the fingerprints, how they find out the fingerprints, because you know that the fingerprints are completely different. So with the help of fingerprints, they'll be able to find it. Similarly, in the crime scene, if there is a drop of a blood, if your teeth is fallen there, or if your semen is fallen, if any part of your skin is getting cut, like, when you're fighting and that person whom you're fighting with has scratched your, mouth, your any part of the body, like hand, and your skin is there in their hand when they're dead, and you are not aware of it. And that case also, they can identify that you are the culprit. Okay, DNA can establish parent-children relation and help us identify individuals from the mortal remains of the disaster sites. Okay, DNA from the blood stain, sweat, and spit from the clothes or soil revert from the crime scene can also be identified the suspect. So experts can also use them to, potential donor for donor organization as well. DNA is very suitable. Scientists have extracted for around 65-year-old human remains preserved only by dissection and cold. Many cold cases have been solved by examining new profile made by DNA extract, and crime scenes material is archived in the archive. DNA fingerprinting is also, helping those people who are accused of wrong convicts. Maybe we have not done the proper thing and there is accusement. In that case also, this is something that can be used. The DNA can be used. Now, next important topic that we have for today here is the glaciers that are there. This is a very, very important prelim space topic. Why? I'll tell you. Because glaciers losing ice at a record level around the globe. According to UNESCO's report State of Cryosphere 2024, cryosphere is a part of the earth that is covered with ice. Okay. So this kind of report that we have here is released on March 21st. Glaciers are melting at an unpredictable rate, contributing to the rise in the sea level. Okay. So what are the records? Glaciers have lost 9 trillion tons of ice lost on the glaciers since 1975 are roughly equivalent to a block of ice, the Germany or the size of Germany with the thickness of 25 meters, right? So these are some of the kind of things that they're trying to tell us. Accelerating five of the last 6 years record loss, greater losses with 450 billion tons of ice that is lost only in 2024 alone. The 2024 is very bad year. In 2024 alone itself, you can see that this much amount of ton of ice is being, dissolved. And because of this, what is going to happen is the sea level is going to be rising. And because of the sea level rise, there will be a lot of other disasters like floods, etc. Water and energy impact. Glacial losses threaten hydroelectric energy and water availability for billions. Mountain communities at risk. 1.15 billion people faces hazards like avalanches, landslides, flash floods. Okay. Eastern African mountains may also lose all the glaciers by 2030, worsening local conflicts are over water. Urgent need actions that needs to be taken here is climate change mitigations are crucial. So these are some of the things that they're trying to tell us that what happens is if this kind of ice is going on. On the same page, there's one more important article that I was not able to cover, and that is related to Kashmir. Okay, an ice frozen for millennium thaws, Kashmir wakes up to the new risk. So a new study that has found is how permafrost in Kashmir, Himalaya would affect 193 kilometers from the road. Okay, so what is happening is permafrost refers to the ground that is remained frozen for at least 2 years. Okay, if this is not at all in the ice itself and it is not exposed to water, I mean not exposed to sun at all, then it is called as permafrost. And the large amount of organic carbon, methane is stored in this permafrost. And if that gets started, if that starts, melting, and all these carbon dioxide and all this methane will start coming out, and this is going to make your climate change most worse again, right? So here we have to understand that the causes are again, of course, rising sea temperature, activities, human activities, etc. So to have a look at it again, this is a mains based topic, right? Thawing for permafrost in Kashmir. In Kashmir also a lot of what is happening here is permafrost, that is the ice that is there on the land for 2 years or more than 2 years. Such a kind of ice is also melting, and this is going to also make our climate change much more worse, right? So now let us see the next important questions for today. What is the primary purpose of DNA fingerprinting? Why do we have to have this DNA fingerprinting? To determine the gender of the individual. Just to determine the gender of the individual, you do not require DNA fingerprinting. So you can determine the gender just by appearance, as well as other things are there. Okay, so there are other things. So just have a look at all the options. Try to find out which one is the right one and try to mention why it is right. Okay, which component of the DNA is used as a different individuals in a DNA fingerprinting? So how do you identify who is different? Okay, is it the entire DNA sequence, genome sequence? Is it the length of the DNA strands? Is it the mitochondrial DNA, or is it the polymerism, or in short, random repeats? So which one is the right option, and why is it right option? If you have any other suggestions also along with this topics that we have discussed, so please let us know that as well, so that we can inculcate that in the classes and give it to you. Thank you so much for your patience.